No, love, I'm just starting to teach, sweetheart. Hi, everybody. Happy Monday and happy last day of class, which is kind of astonishing. Okay, so first up, anyone that has any questions about material that we've covered so far? This is really your chance to clarify stuff, go over things before the exam on Wednesday. So talking about anything that you have questions about, please ask. Either in the chat or unmute your mics, either one. Otherwise, it's going to be a very short class otherwise, which is fine, but that would be a missed opportunity, I think. The exam is over PowerPoints. Which one's this? 18? Yeah, 17 and 18. So this one. Exam is this week. It's on Wednesday. At least according to the schedule. That's it's on December 1st, which is Wednesday. So back to Infinity's question, the exam is largely over PowerPoint 17. So control of gene expression in prokaryotes, which is kind of largely on the general principles of control of gene expression. The whole inducible, repressible, negative, positive, that kind of stuff. Lac operon, trip operon, right? In quite some detail, because we spent a lot of time on those, right? And as you know already, the number of questions on something is directly proportional to how much time we spent talking about it. And then on the control of gene expression in prokaryotes. And likewise, there's a lot of stuff here, right? A whole 36 slides of information that we have covered over several class periods. And then the final bit that, that we'll talk about is CRISPR-Cas9. Not going to really talk about, that's where the transgenic alligator embryo is. Not really going to talk about much else in here because of both of time and also because we didn't have time to cover much of this in the other course section, and I'll try and make sure both of these sections are treated the same. So that's pretty much it. So really just 2 and a bit PowerPoints, 17, 18, and a little bit of 19. Oh, not 19, 20, sorry. I can just about hear you. Who's that?
Okay. I think I have a question.
Sure.
For trip operon expression during attenuation, I understand we have the two different ways that the regions can bind. So specifically, I think it was for when 2 and 3 bind, it still allows transcription, but I still kind of don't understand how that loop affects the ribosome. So is it just transcribed and then the ribosome, like the loop doesn't stop the ribosome? It's still able to like go over it and continue later? Or is it transcribed and then it just chills out waiting for some other time?
So the key point there is that Bloody hell. Sorry, my dogs are eating their breakfast and making a racket doing so. The ribosome, it doesn't matter that it's stored there, right? Because this protein doesn't do anything. So where's the, each of these structural genes has its own start and stop codon. So what happens to the translation of the leader region doesn't affect the translation of the other regions, right? So this ribosome, it's just for that one short stretch. So really its purpose is not to translate anything. Its purpose is to detect the level of tryptophan. The key point here, though, is that this loop, when it forms, is far away from the RNA polymerase, right? So it does not interfere with the RNA polymerase, and so the structural genes can be transcribed. The fact that the ribosome is just kind of hanging out there rather forlornly doesn't have any effect at all. All its job is to do is to prevent, well, essentially to make sure that region 2 binds with region 3. And it does that by pausing on region 1. So it could hang out there for as long as that mRNA is in existence for. It really doesn't matter. Its purpose is purely to detect whether or not there's enough tryptophan present or not.
Okay.
Does that make
answers my
sense?
question. I was getting real tripped up on that ribosome, and I don't need to.
No, no, and it's funny because that's something that I've learned that trips students up actually this semester, which is pretty neat. So yeah, that whole leader region, that protein doesn't do anything. It's purely being made to detect tryptophan levels.
Okay, I have another question.
Go for it.
For the lac operon, it's inducible, and I understand the inducer part of it. The inducer binds to the repressor, but that repressor is not the same thing as aporepressor, corepressor. Like that's just a repressor in and of itself, and it's not the same thing.
That's correct. So if you ever come across something called an aporepressor, that tells you kind of inherently it needs a corepressor to bind DNA. Simply the apo part at the front of it tells you without anything else present, it cannot bind DNA. On the other hand, if it's just a plain repressor, which is what LAC-I is, pardon me, then it can bind DNA in the absence of anything. So that's what is going on here. So repressors bind DNA in the absence of anything else. Not a very elegant way of putting it, but it'll do. Aporepressors, e.g. TRP, actually is LAC-I. Quit it. Sorry, dogs again. They're just being furry nuisances this morning. So they require, aporepressors require the presence of corepressor.
Okay. I think my last question was in eukaryotic expression. I understand there's post-translational modification, and then there's a bunch of things that happen before translation. But was, as far as like labeling things that we've covered, was there anything that happened during translation?
Not really, no, because sorry. Quit it.
Okay, so I was
No,
muted.
hang
I
on.
was trying to ask a question and I'll ask it again because I was muted.
Sorry,
So
I
for
didn't hear that last bit.
eukaryotic gene expression, I understand there's post-translational modification. There's a lot of things that happen before translation, but as far as things that we covered, was there anything that happened during translation for gene expression control?
So yeah, sorry, I was distracted by my younger dog being a complete pain in the ass. So if it looks like I'm not paying attention to you, it's not because of you, it's because of this thing. Okay, so in terms of control of gene expression, one thing to really, what kind of control is all about, at least in terms of transcription and translation, typically whether or not it happens. Once it actually starts, once transcription starts in eukaryotes at least, and translation starts in, I think both really, but definitely eukaryotes, it just occurs, right? There's nothing really that stops it. So all of the control, the kind of sentence around translation is all about whether or not it actually happens, right? And so where's, what slide did I want to talk about? So one way of controlling that is this microRNA pathway. Let me just, come on. This one here. There you go, move it that way instead. So you can inhibit translation of an existing mRNA by having a non-coding RNA bind to typically the 3' UTR, right? So that's what's going on here. So if you want to say transcription of a gene has already occurred, but now you want to shut down translation of that gene, of that mRNA, sorry, then typically you would express another RNA, a non-coding RNA, which is used to shut that mRNA down. And so typically in terms of translation, that's done by inhibiting translation. And so this RISC and guide RNA complex that we got down here stops the, typically it stops translation by stopping the 3' end from bending around and interacting with the initiation complex. Another way in which translation can be inhibited is if there's no mRNA there at all, right? And that is by the small interfering RNA pathway. And so it's the same kind of principle in that a non-coding RNA is expressed from another gene, right? So this is a separate gene from the one that we're talking about the control of. And if the matches are good enough, then that RISC complex and guide RNA actually degrades or cuts the mRNA, which results in its degradation. So really, you're kind of on the right track because translation itself, nothing much happens there. It's either before it in terms of initiation of translation or even the ability to translate that mRNA, right? Because if it's not there, you can't translate it. Or after translation, where it's all about these post-translational modifications. And actually in eukaryotes, it's the same with transcription, right? It's all about whether or not this basal transcription apparatus assembles and is activated on the promoter. And so if it is, you get transcription. If it's not, you do not. And then, I don't know where she's going. After transcription, then it's all about RNA processing and RNA stability, essentially. So it's kind of the bit in between transcription and translation is where, you know, that other level of control of gene expression occurs. Yeah, and one thing to get across as well is that, you know, the RNA polymerase and the ribosomes, they're dumb as a box of bricks. You know, they don't know what they're supposed to transcribe or translate or how much. They simply assemble, and if they're told to go, they go, and they'll just keep going until they stop. That's pretty much it. All of the control is about other proteins or non-coding RNAs and proteins that control whether or not that happens. The actual RNA polymerase, the transcription process, the translation process, like once they start, they continue. And then they finish, right? So it's all about controlling whether or not they actually start or not. Does that make sense?
Yes, I don't think I have any more questions. Thank you.
Awesome questions. Thank you. But there is an awful lot going on in eukaryotes, for sure. And another point to kind of stress is that these are all the different possibilities that we're talking about. It doesn't, which ones apply to any particular gene is very much dependent on that particular gene, right? There are just a huge number of potential combinations.
Oh, I lied. I have one more question.
No worries, go for it.
As far as like, excuse me, like the case studies that you showed us, and then like the bit polydactyly,
Uh-huh.
is that going to be on the exam, or were those just used as examples to highlight your points?
Those will be on the exam, maybe. Sorry, I shouldn't joke about exam questions. No, those are part of the content which may be on the exam.
Okay.
Yeah. And really, I guess at the end of the day, what I want you to get from them is not just this kind of anodyne sort of, you know, proteins are present or absent, you know, or cis-regulatory elements are present or absent. What I really want to get across with the polydactyly one, sorry, is that those elements functioning has a huge impact on organismal level function. Right, so these are small bits of DNA, but their present or their function or lack of function can have really large functional consequences on the organism itself, right? So these are just small bits of DNA, many, many bases away from the gene itself, but changing them can have really quite marked physiological differences. And similarly down here, the points from these is that, I mean, actually, even this is the most remarkable one, really, of the two examples, because all you're doing is adding or removing a phosphate, right? So that's like 4 or 5 atoms, I think, from a protein. And in doing so, you completely change the function of that protein and its localization, which is in many cases tied to function. Right, so phosphorylation is by far the most common and complex protein modification there is, right? And it has really huge consequences. So that's kind of the main point from that one. And from this one, it's really about 2 things. One, cleavage, and 2, a cascade of events, right? And so this is actually a very neat picture, which is why I included it, that kind of physically shows that cascade, or graphically rather shows that cascade, right? And so it's all about control. You want to be extremely precise about the control of blood clotting. Because if you do it when you don't need to, you die. And if you don't do it when you do need to, you die, right? And so you can see that there's, as I often go back to like evolution and the like, there's an extraordinarily strong selective pressure maintaining this system, or that even led to this system, right? Because if it works inappropriately, consequences are essentially fatal, right? And so that's why, and also when you do need it to work, you need it to work very quickly. So that's why we have this cascade, right? You require at least 3 different things to work for the whole process to actually function. And 2 of those are by cleavage, right? Cutting of proteins, right? So those proteins don't work until they're cut, until they're separated from, other bits that are attached to them. And also because we have, one thrombin molecule can actually catalyze multiple fibrin molecules and so you can have this kind of spreading out effect. It's like a river delta, right? One thing can do 2 things, each of those can do 2 things, and so you have this very quick expansion of activity. So yeah, those are most definitely exam worthy subjects, without a doubt, because they illustrate really important points that I want you to know and understand. Good questions, thank you. Love questions. Anybody else? Feel free to pipe up. No stupid questions whatsoever.
So cleavage is just not also just referring to the cells or themselves, but it also to the proteins and its structure and shape and how that affects what it does.
Absolutely. That's really exactly what it's about. And so the key point is that I love using these circle things. This protein does not stick to itself. And so you have flown around in your blood a whole ton of fibrinogen, right? Which doesn't interact with anything else, including itself, right? That's what we mean by it being soluble. The only way that it can stick to itself is if you cut off these wibbly bits called A and B, right? Once you do that, you have something called fibrin, right? Changes it from, it's still the same, essentially the same protein, just missing some bits. And that fibrin, come on. That sticks to itself and forms or helps a blood clot form, which stops you from bleeding out, basically, right? Even from a fairly minor scratch. If this didn't happen, even actually yesterday, I'm always getting myself cut and stuff. Managed to stab myself in the toe with a piece of metal, right? If I was a hemophiliac and I didn't have access to various drugs to help me, that I would have died from that, right? It wouldn't have just been a, ow, shit kind of moment. It would have been a, dead on the floor of my garage kind of deal, right? And so you really need this to happen. You need to happen very quickly too, right? But you also don't want this flown around in your blood. So the thing that controls that switch from fibrinogen to fibrin is thrombin, right? And that switch is by cleavage. So thrombin is an enzyme, right? It actually cuts very specifically little bits off of fibrinogen. Now, thrombin itself as an enzyme is only active when it is cleaved from another bit of itself, essentially, right? And so you also have in your blood at the moment prothrombin, which is this protein, right? And it is inactive and is unable to recognize fibrinogen and cleave it. So the thing that really starts all of this whole process is the release of these clotting factors. And those are one, can't remember which one it is. No, I can't remember off the top of my head. Absence of one of these is what causes hemophiliacs to be unable to do this whole process. Right, and so those clotting factors, in ways I'm not 100% familiar with, cleave prothrombin to get out of it. Thrombin, that thrombin is then able to work. I just got to send this to the back. It's just pissing me off. There you go. And then thrombin recognizes fibrinogen, turns it into fibrin. Fibrin sticks to itself and makes a blood clot. So these proteins, prothrombin and fibrinogen, are circulating in your blood, right? You don't change the levels of those proteins. You don't upregulate gene expression, right? So you don't make more of those proteins to have in your blood when you have a blood clot, because that can't happen quickly enough, right? It takes time, 30, 40, 50 minutes, let's say, to be able to produce enough of these proteins for them to have an effect. And that's too long, right? And so you can't wait for that to happen. And this is actually, it's a good question, right? Really, because it comes back to another point, which I don't think I really stressed, not because it's unimportant, but because I forgot. And that point is that some things that you need instantly, if you need them instantly, you can't wait for the whole process of transcription and translation to occur. And so for those kinds of activities, and again, always coming back to insulin is another one, you have to have some means of storing that protein or having it in your body already made, and then activating or getting it to where it needs to be active, like on command, like on a signal, right? And so that's a great example of why post-translational modifications are so important, because there's a lot of things, cell signaling, enzymatic cascades, cell signaling, right? Various things where you have to have that protein present, but you don't necessarily want it to be active, right? And so post-translational modifications are really about that, right? We're not changing the level of physical protein, we're changing the level of active protein, right? Does that make sense? Yeah, that's kind of what post-translational modifications are all about. Any other questions? If you do have any, just pipe up and ask. I'm happy to stop and go back. Okay, so the last thing that I want to talk about is just something cool and actually very scary, and that's CRISPR-Cas9. Actually, this should have a 9 after it. There you go. Pardon me. And CRISPR-Cas9 is actually had a friend who works on this. I think she's at, where is she at now? Emory? Oh, this will be on the exam, Caitlin. Maybe, not in a huge amount, but it will be there because I'm going to be talking about it. So I want to make sure there's enough time to cover it. Obviously, we're only going to be covering it for at most 18 minutes. So you're not going to have like 15 questions on the exam on CRISPR-Cas9. But it probably will be on there. So yeah, she works on CRISPR-Cas9, but she works on it in the kind of the original purpose, right? And so the original purpose is it's a form of bacterial immunity, which is kind of a cool concept, right? Because we don't really think of bacteria having an immune system. But essentially bacteria are plagued by viruses, right? If you're a humble bacteria, you know, wandering around, absorbing nutrients and stuff, your main foe, other than unfriendly other bacteria and nasty and vicious yeast or fungi, which are actually really, really nasty to bacteria. That's where we get almost all of our antibiotics from, for example. Your other worry, if one were to worry as a bacterium, would be viruses. And so CRISPR-Cas9 is all about maintaining a memory of what viruses you've been infected by, right? Obviously not just you as a bacterium, like your, you know, predecessors in your bacterial family tree. And they do that by, and that's what CRISPR stands for, something, 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 palindromic repeat. I can't remember what all the letters stand for. It stands for something, I just can't remember what. Concatenated, anyway. Essentially what the bacterium does is it takes a little snippet of the viral genome and it sticks it in its own genome in a particular set of repeats. And so if that bacterium is infected by that virus again, it's able to recognize it and destroy the DNA of that virus, which is pretty cool. Okay, so how does that work? And this is where it gets that's a really good question. So just kind of think about this for a second. Two reasons why not. One is that COVID is an RNA virus, and I think this only works against DNA viruses. Don't think bacteria are infected by RNA viruses. Not 100% sure, but I don't think so. So that's one. The second is that this is too complex of a process and too difficult to get inside cells. So the reason why it's would be difficult to get inside cells, is because of the Cas9 protein, this thing here. And so the COVID mRNA vaccines work by, they're actually really cool. They're short pieces of mRNA that are encapsulated, which is a cool word in and of itself, if I can spell it, in small lipid vesicles. Vesicles. And a vesicle is basically a little bit, it's like a bit of, it's a ball or consistent of lipids, similar to your plasma membrane, essentially. And so those lipids allow the vesicle to be absorbed by your cells. And this is actually the most groundbreaking part of the COVID mRNA vaccines. It's getting the mRNA into the cell. It's not the mRNA itself, it's the actual delivery mechanism. And that's also why these vaccines have to be kept cold, right? Because otherwise they fall to pieces. And that having those lipids, vesicles absorb, then allows the mRNA, which encodes a piece of the COVID, I think it's a spike protein, would be my guess, bit on the outside of the virus, to be translated. Then because that protein is foreign, it's basically stuck on the outside of the cell. And there are actually specific cells called antigen presenting cells in your body, which this is their main job. I'm not sure that those are the target, maybe it's other ones. I'm not 100% sure. But anyway, that's then recognized as an antigen. And that gives you immunity. So getting RNA into the cell is not a trivial endeavor, right? That's really basically the Nobel Prize winning breakthrough of the COVID vaccines. Because we've never had an mRNA vaccine before, right? It is literally game changing, right? It changes everything in terms of vaccine development. However, proteins are very difficult to get into size cells because they're big. They're very big, and it's just not easy. So that wouldn't work this wouldn't work very well as an anti-virus mechanism. Now, let's talk about actually how this works, right? So this is a kind of endogenously, right, in its normal natural context. This is a means for destroying bacteria viruses, right? So viruses that infect bacteria. What the, again, Nobel Prize winning change is that, what's her name? Something Doudna? I can't remember the other lady who won the Nobel Prize. Anyway, what they and some others recognized is that this is a very specific way of targeting DNA, right? And it's extraordinarily specific. So the specificity comes from the guide RNA. The Cas9 protein is really just the effector. It's what does something. And so the name of the game is that you can design, and there are actually programs that will do this for you, or even, I think, browser apps. You can design a guide RNA to almost any sequence you wish. Right? That's where the power of CRISPR-Cas9 system comes in. Right? You decide, right? And so if you want to target a gene or a piece of DNA, as long as there's one of these PAM sequences, which are short, I think they're trinucleotide sequences. I don't know why, they're just required for Cas9 binding in some way, I'm not 100% sure. But as long as it's somewhere near one of these, what you do is you design the guide RNA sequence and you have it synthesized for you. And that sequence is going to match the bit of DNA that you want to get cut. So this guide RNA is what directs the Cas9 or the effector complex to that bit of DNA to cut it. Because that's what it does normally in bacteria. But now instead of going by, old viral remnants, right? Now, if say you want to, oh, hey, here we go. It's just a fun and slightly scary idea. There's a mutation in cattle. Is it Belgian something cattle? They are hyper muscled, right? And so they look like basically Arnold Schwarzenegger cows, right? Like super ripped, bulging muscles. They're really quite disturbing, to be honest. Anyway, what you could do is say you wanted to make a super ripped like baby. You could design that guide RNA to the gene required, which is something it's like myosin or myosin receptor, something that controls how much myosin, which is one of the components of muscle cells. Right. Now that then gets injected into the embryo along with the Cas9 protein, which you make separately. And those form an effector complex. And what they're going to do is they're going to scan much the same as the RNA-induced silencing complex does, right? They're going to scan the genome for a match, right? And so where are we at? RNA-induced silencing complex does that for mRNA. So where it finds a match, it cuts the mRNA. For CRISPR-Cas9, it does that, but for DNA, right? So when it finds a match, the CRISPR-Cas9 kind of unwinds the DNA a little bit and then creates a double-stranded break. Creates double-stranded breaks. And those are catastrophic. So if you have double-stranded breaks, and that's actually what ionizing radiation like gamma radiation from nuclear explosions and stuff does. If you have double-stranded breaks, you have to fix them. Even if you do a shitty job, you have to fix those double-stranded breaks. Otherwise, that cell is gone. It's toast, right? And so the way of doing that, right, in the absence of anything else, is called non-homologous end joining. I'm just going to shorten that to NHEJ because it takes too long to type out. And basically, I call this a Hail Mary DNA repair mechanism. For DSPs. And it's not a very good one, right? So, and the problem is, right, if you have two ends of DNA flapping around, oh, you're just gonna have to wait. How do you know which end goes with which? Right? That's the kind of the problem that cells face. And so non-homologous end join is basically like, these look about right, stick them together, right? Whether they are or not. It's just like double-stranded breaks are even worse. And so, and almost always introduces mutations. Either short insertions or short deletions. And so essentially what happens is, say you want to knock out like that particular gene, all you have to do is cause a double-stranded break in that gene so that when it gets stuck back together again by non-homologous end joining, then there's a mutation there. And as we've known, right, if you target that break to the coding region, the addition or subtraction of bases, particularly if they're less than, if they're not in multiples of 3, right, because of the whole codons and reading frame deal, then you'll have a truncated nonsense protein, right? You'll cause a catastrophic frame shift mutation. Right. So that was kind of how CRISPR-Cas9 started, right? Just as a basic mechanism of let's screw up this bit of DNA in a very targeted way, right? We can screw up DNA till the cows come home via lots of other mechanisms. Just UV irradiation, for example, will mess them up, right? But what we haven't been able to do until CRISPR-Cas9 is to do this in a targeted way. That's what makes it so scary, right? And then as, you know, people got hold of this technique, clever minds started churning and thinking, okay, how can we improve this? And so the next step is now, rather than just causing some mutation of some sort, if you provide DNA at the same time, so now you got guide RNA, the Cas9 protein, and some donor DNA, if that donor DNA matches either side of that break, so you can have something funky in the middle, say a GFP protein, but if the DNA either side of that matches either side of where you're going to make the break, now you can have homologous recombination. And cells will always choose homologous recombination to fix double-stranded breaks if other DNA is present. So remember, we talked about this way back when. That's going to be during cell division normally, right? Because you have your copied your sister chromatids and your homologous chromosomes around. But if you provide that, then the cell will go, whew, don't have to use that crappy non-homologous end joining, and it will actually swap in your donor DNA. And so now you can insert DNA into regions of your choice. So say you wanted to tag a protein with GFP to see where it's expressed. Now you can actually insert a GFP or green fluorescent protein gene and put it in frame with your target gene. So you can create those fluorescent protein fusions, like the ones I showed you about my own work, but this is actually in the genome of the organism. And the scary thing is now you can create any change you want. And really, you can do that to anything that you want, right? And so there's actually been, this is very much a Pandora's box, right? There has actually been a case where it was in China, a researcher actually created genetically modified people, two girls, which he genetically modified to have a mutation that made them resistant to HIV. Right. Obviously, there's huge condemnation, but now the kind of cat is out of the bag. So if you want to make genetically modified people, now you can. So anyway, leave it on that note. Future bioethics class would have to go through that in more detail. So this is just probably the biggest scientific advancement in, or at least 20 years, right? You can do whatever you want, Caitlin, if you have the ability to actually do that, and a source of embryos, fertilized embryos to work on. There's nothing to stop you other than your own ethics and your fear of being punished, essentially, by your own government. So potentially, if you can figure out the different developmental pathways required for scales to grow, you could have scales. If you want to make super muscled humans, we know how to do that in cattle. You can do that in humans now, right? Obviously, you know, it is insane. And it's insane because you don't know fully the consequences of making those changes. And also the individual being changed doesn't have a say in it, right? Because it happens at the level of the embryo. So you could be born and have like some change made to you, and you have no say in that whatsoever. It's in your DNA. You can't change it. Yeah, it is arguably most powerful. Exactly, designer babies. Watch Gattaca. Actually, if you haven't done it already, it's a really, really cool. Ethan Hawke and I can never remember her name. The actress from Kill Bill. Yeah, it's very interesting. Now, obviously, there's a lot of, pressure not to do that stuff, but that's a societal thing, right? The actual technology is there. It's just whether or not we decide to use it. All right. Anyway, that's enough from me. I've gone over a few minutes already. Leave that humdinger with you. I will be in Zoom on Wednesday. If you have any last minute questions, I'll be there for most of the class period from 9 o'clock. Exam will open at 9 and it will be open until midnight. All right. It's been an absolute hoot, everybody. Thank you for hanging around and being part of it. And I'll see you sometime soon.